Good morning student. Today I will teach you asexual reproduction in unicellular organism. First of all, I explain you what is the means of reproduction. Reproduction is the biological process in which new offspring is formed by the earlier existing organism. Method of reproductions, there are two methods of reproduction. First one is the asexual reproduction and second one is the sexual reproduction. Now I define you asexual reproduction. Asexual reproduction is the process in which new organism is formed by the earlier existing organism of same species without involvement of a gamete. So what's the means of that? Asexual reproduction is the uniparental process. In this type of reproduction, a single organism is participated. Because of there is no involvement of a gamete, so no genetic recombination we can see in the new organism. And the drawback, this is the drawback, because in this process we cannot find out or obtain a new genetic recombination. But we can find out without a very short period of a time, we can get a more amount of a new organism. So it is a fast process. This is the advanced features of the asexual reproduction. Now I explain you asexual reproduction in a unicellular organism. In unicellular organism, asexual reproduction is occurs by three methods. Okay, which are they? First one is the binary fission, second one is the multiple fission, and third one is the budding. First I will explain you binary fission. Binary fission, this is made up of two words. Binary means two and fission means splitting. When a single organism is split and form two new organisms, that is known as the binary fissions. Binary fission generally occurs in prokaryotes, protista and eukaryotic organelles. Such as mitochondria and chloroplast. I think everyone understood. Okay, binary fissions means splitting. Bi means two and fission means splitting. So this is occurs in the prokaryotes, protista and eukaryotic organelles. Okay. So what we can see in this process, a parental cell is divided mitotically by two similar daughter cells. The binary fission is depend upon the division of binary fission is depend upon the axis of a organism. On the basis of axis of a organism, binary fission is again divided into a three types. Okay, binary fission is again divided into three types. Which are they? First one is the simple binary fission. Simple binary fission example is a amoeba. Second one is the transverse binary fission. Example is the paramecium. And third one is the longitudinal binary fission. Example is a euglena. Okay, first of all I explain you simple binary fission. In the process of simple binary fission, what is the example of simple binary fission? Amoeba. All of you know amoeba don't have a specific shape. So there is any particular axis by which we can divide them into two equal parts. Because of that binary fission is known as the simple binary fission. So what happen? Actually binary fission is occur in the favorable condition. This is the fully grown amoeba. After the full growth, the amoeba undergoes mitotic cell division. In this process, first of all, the process of karyokinesis is caused. In the process of karyokinesis, the nucleus is divided. In the first step of a nucleus, they are elongated, and after that, the division is completed and nucleus is divided into two equal parts. Simultaneously, the process of cytokinesis occurs. In the cytokinesis, cytoplasm is started to divide. And after the division of cytoplasm, we can get two daughter amoebules. What we can get? We can get two daughter amoebules. So which process occurs here? Here the process of nucleus division. And here is the process of cytoplasmic division. Okay, I think it is clear for you. And what is the result? We can get a two daughter amoebae. Now second one is the transfer binary fission. So we all know what is the shape of paramecium. Paramecium is the slipper shape. Okay. So what we can find out here? This is the shape of paramecium. How many nucleus we can get? Two nucleus. Okay. And all of you see, one is the smaller in the shape as compared to the other one. And what we can see here? We can also see a presence of a oral groove. What we can see here? Presence of a oral groove. Only on the basis of presence of oral groove, we can decide the axis, that is the transverse. In this process, what happened? First of all, same condition is occurs here, karyokinesis and then cytokinesis. But simultaneously, who is also started to divide? Your oral groove. Your oral groove is also started to divide with the nucleus. There is the elongation in the process of a nuclear division. Same condition with the macronucleus. So because we can see two type of a nucleus. This is the micronucleus and that one is the macronucleus. As the process of karyokinesis is completed, so nucleus is divided into two equal parts. Okay. After that, which process are going on here? After that, we can see the process of a cytokinesis. Okay. In cytokinesis, again here is the formation the constriction ring, and we can get a two paramecium. Okay. How many paramecium we got? We can got two paramecium. That is the daughter paramecium. So karyokinesis is occurred, then cytokinesis, and what is the result? We can get the daughter paramecium. Only on the presence of oral groove we can decide their axis transversely. Because of that, this fission is known as the transfer binary fission. And which type of a cell division is occur here? Mitotic cell division is occurs. Okay, now the third step we are taking. What is the third type of a binary fission? Longitudinal binary fission. Longitudinal, I think the word is a explain you longitudinally division. So what is the example? Euglena. So just as we can see the structure of a euglena, in this structure we can see the flagella. How many number of flagella is here? Only single. And in between we can see the nucleus. So if we can divide the euglena into two equal parts, so which axis we have to take? Longitudinal axis. In this process, first of all, your flagellar region is divided into two halves. Okay, and simultaneously which process are going on? Division of a nucleus. After that, this furrow is a dipper and the euglena is divided into two daughter euglena. So on which axis we can see? Longitudinal axis. Because of that, this division is known as the longitudinal binary fission. Okay. Now, binary fissions occur in a favorable condition when there is a sufficient amount of a food are present. Okay. So on the basis of axis, we are divided the binary fission into three types: transfer binary fission, simple binary fission, and longitudinal binary fission. Our next topic is the multiple fission. Okay. Multiple fission, it occurs in some protista, just like amoeba, paramecium. Okay. And multiple fission occurs in the adverse condition. So this is the way by which they can exist their species in the environment. So what happened in the multiple fission? The parent amoeba is divided into so many daughter amoebules within a time. Okay. So in the process of multiple fission, what happened? First of all, what is the shape of amoeba? We know about that have an irregular shape. And this elongation process is known as the pseudopodia. So in the first conditions, what happened? Amoeba withdraw their pseudopodia. When they withdraw their pseudopodia, they become a round in a shape. And after that, they make a hard covering around them. And that hard covering is known as a cyst. What is the cyst? It is the protective layer which protect the amoeba in adverse condition. At that time, the nucleus which is present inside the amoeba divided mitotically and form so many nucleus. So now the amoeba is converted into many nucleated structure. At the same time, the cytoplasm is divided and covered the nucleus, means make a new amoeba. Okay. So inside of a single amoeba or inside of a parent amoeba, we can see so many amoebules. After that, what happened? As the favorable condition is occurs. So this is the division of nucleus and cytoplasm. First of all, nucleus is divided. After that, cytoplasm is divided. As the favorable condition is come, the cyst is a burst or a rupture, and the daughter amoebules which are present inside of the cyst comes out from this cyst. In this way, they exist their number in the environment and exist their species. Okay. So, this is the method of a multiple fission. Now, the third one is the budding. In unicellular organism, budding occurs in a fungi, unicellular fungi. In fungi, what we can see? The parent fungi fully grow. When the parent fungi is fully grown, we can take the example of a yeast. Yeast is a fungi. So, what happens? The bud is formed. What is the means of bud? In the parent yeast, a outgrowth is formed, and that outgrowth is known as a bud. What we call this lesion? Bud. This is the bud, outgrowth, that is known as a bud. After that, what happens? The parent nucleus is divided. As the parent's nucleus is divided, it moves into that. We can just show here the division of nucleus. Okay. So, nucleus is divided mitotically and moves toward the bud. After that, they take the all nutrition from the parent yeast, and when they are sufficiently grow, they detach from the parent's yeast. They detach from the parent's yeast and make their colonies, and they are independently live their life. Okay. In this way, what we can see? We can see the so many daughter yeast. Okay. What is this one? This is the daughter yeast, or what we can say? This is the colony of yeast. So, what we can study? Asexual reproduction in unicellular organism. We study the three methods: binary fission, multiple fission and budding. Thank you.